Hello everyone. In a previous video we have studied about the Griffith's experiment of transformation, and we have also studied about the conclusion of Griffith experiment, how this experiment was carried over by three scientists, Avery, McLeod and McCarty. In today's video we are going to study about another experiment performed by Harshey and Chase, and this is called as the Harshey and Chase experiment. In Harshey and Chase experiment, they used two organisms. One is the T2 phage, the other one is the E.Coli bacteria. They used the T2 phage and the bacterium E.Coli. So, first, before going into the experiment, let's see how this T2 phage infect the bacteria. There are four important processes of infection. In the first process of infection, it gets absorbed to the surface of the bacterial cell. The T2 phage gets absorbed to the surface of the bacterial cell. In the second step, it injects its DNA. In the third step, the DNA which has been injected replicates. In the fourth step, the bacteria undergo lysis and the T2 phage is released out. And let's see the structure of the T2 bacteriophage. It consists of a protein capsid which is polygonal type, and this polygonal protein capsid consists of a DNA which is said to be the genetic material. It has a column which is surrounded by a protein sheet, and it consists of an end plate, and from this end plate there arise the end tails. So we are mainly concerned with this polygonal protein capsid and DNA which is at the center. Now this bacteriophage insects the bacterium by these four processes. First, it gets absorbed on the bacterium. Next, it injects the DNA inside a bacteria. The injected bacteria undergoes a replication. In the fourth step, the bacteria undergo lysis, that it breaks open to release the multiplied T2 bacteriophage. Now, in this experiment to prove DNA has a genetic material, Harshey and Chase used two types of T2 bacteriophage. The one is called as sulfur 35, the other one is phosphorus 32. These two substances are radioactive substances. These two substances are radioactive in nature. One is sulfur 35, the other one is phosphorus 32. Why they use sulfur 35 is that they thought that protein may be the genetic material. So, if protein is the genetic material, then sulfur gets incorporated into the protein capsid. This is because we know that the sulfur is mainly present in protein but not in DNA. We know that proteins are made up of amino acid. The amino acid cysteine and methionine mainly consist of sulfur. So, if sulfur 35 radioactive substance is present and if it binds to the protein coat. If phosphorus 32 is a radioactive substance present, we know that phosphorus is an important component of DNA material. This phosphorus 32 mainly binds to DNA but not to proteins. Now, they allowed these two bacteriophage, that is radioactive bacteriophage, to infect the E.Coli. So, they observed two different things. One is the bacterium. Now, once the infection, we have the bacteriophage infecting the, this is containing sulfur 35, the other bacterium which is being infected with phosphorus 32. Now, once it is been absorbed, it injects the DNA. Now the DNA gets injected. Now once the DNA gets injected in this first case, we know that the sulfur 35 is mainly present in the protein capsid but not in the DNA. So the DNA is non-radioactive because it does not contain any radioactive substance. In this case we are only injecting sulfur 35. Whereas in the second case the DNA is being coded with phosphorus 32 because the DNA contains phosphorus substance in it. So phosphorus radioactive substance get incorporated into this DNA. Now this DNA which is being injected inside is radioactive in nature. Now this is centrifuged. So we have the test tube here. Now both the things are centrifuged. When we centrifuge, we get pellet and supernatant. This is the pellet. This is the supernatant. The substance which is heavy settle at the bottom of the test tube, and the one which is lighter floats up, and this is the supernatant. Now here in the pellet, the bacterium containing the DNA will be at the bottom. Here, even here, the bacterium containing the DNA will be settled at the bottom, and once the DNA is being injected into the host organism, now this capsid becomes empty. The empty capsid is called as ghost. Now we can see only the ghost here. Even we have the ghost here. Now the pellet and the supernatant are analyzed. In the first case, in this case, when the supernatant is analyzed, we know that the sulfur 35 contains the radioactive substance, that is the protein capsid contains the radioactive substance. Here it is in the protein capsid. So when the protein capsid is in the supernatant, when the supernatant is analyzed, the capsid is present and it contains the radioactive sulfur 35. Whereas when the pellet is being observed, the DNA does not contain sulfur 35 because this bacteria was mainly infected only with sulfur 35. So the DNA does not contain any sulfur 35. So here there was no radioactivity. Coming for this part, so analyzing the pellet and the supernatant. Here the supernatant consists of a protein capsid, that is the ghost, without any radioactivity, whereas the pellet, when analyzing the pellet, the dense bacterium, the host bacterium contained the radioactive phosphorus 32. They could find the radioactive phosphorus 32. So they came to the conclusion saying that since phosphorus 32 is being incorporated into the DNA, now once this is being injected, it undergoes, once this is injected, the next step is the replication. The DNA undergoes multiplication. Here there was no multiplication because in this there was multiplication, but after this there was no sulfur 35. In this all the DNA material contained phosphorus 32 in the DNA. So after that there is a release. After once there is infection, we have the replication. After replication, we have the lysis of the bacterium, and there is a release of this T2 phage. Now in this case the T2 phage consists of phosphorus 32, whereas in this case there was no sulfur 35 in the T2 phage which was released. So from this they came to the conclusion saying that DNA is a genetic material.